Hello, I'm Zhongliang Zu from Vanderbilt University Medical Center. Today, I'm going to present our paper entitled MR Imaging of a Novel NOE-mediated magnetization transfer with water in rat brain at 9.4 Tesla. MT and CEST can provide molecular imaging with relatively higher sensitivity than MRS. Previously, set from AMAT at 3.5 ppm, amines from 2 to 3 ppm, and related NOE at minus 3.5 ppm have been reported. Recently, we found a new NOE-mediated MT signal at around minus 1.6 ppm, named NOE minus 1.6 here. In this paper, we show how to specifically quantify this signal, its distribution in rat brains, application in tumor, and possible molecular origin. In CEST imaging, a Z spectrum is usually obtained, and an asymmetric analysis named MTR-ASIM is typically applied to isolate CEST effect from other non-specific factors. This method works well in simple model simulations of phantom experiments, as shown in the upper two figures. However, in biological tissues, there are both downfield CEST and upfield NOE effects. As a result, the MTR-ASIM makes these two effects. To solve the problem of MTR-ASIM, a Lorentz difference analysis was developed to remove background signals. The dashed line in the upper figure shows the fitted reference signal. The lower figure shows the CEST radio spectrum. Note that both downfield CEST and upfield NOE can be shown in the CEST radio spectra. However, recent studies by Zeiss indicate that MTR cannot fully remove the background signals. Instead, an AREX method is more specific than MTR. This figure shows the AREX radio spectrum. Note that the NOE minus 1.6 become more pronounced than that in the MTR spectrum in the upper slide. This figure also shows another problem, that the NOE minus 1.6 overlaps with the nearby NOE at minus 3.5 ppm. Here, we also performed a two-pole model fitting of AREX radio spectrum to isolate these two peaks. In this study, 9 healthy rats and 8 rats bearing NOE tumors were scanned to evaluate the characteristics of this signal in tissues. Reconstituted phospholipids and cultured cell lengths were also measured to evaluate the possible molecular origin and contrast mechanism of this signal. We first optimized the NOE minus 1.6. Here is our results on rat brains with 3 different powers. Note in the right figure that this signal is optimized at 1 megatesla. We then quantified the NOE minus 1.6 signal acquired with 1 megatesla. Here is the AREX radio spectra and the fitted NOE minus 1.6. Note that this signal can be isolated from the other effects. We studied the distribution of the NOE minus 1.6 in healthy rat brains. The left column shows the autonomy, maps of the fitted amplitude, offset, and line width of this signal. The right column shows the Z spectrum from different rat brain regions, and the statistical analysis of these parameters. Note that the offset and line width vary for different regions. We also evaluated the application of NOE minus 1.6 in tumors. Figure A shows the NOE minus 1.6 map. Note that the tumor can be clearly found in this map. Here is a statistical analysis of NOE minus 1.6. Note that its amplitude shows significant contrast between tumors and contralateral normal tissues. We also performed experiments on reconstituted XPC to evaluate the possible molecular origin of the NOE minus 1.6. Note in Figure A the presence of the signal in Z spectrum from XPC. Also note in Figure D that this signal is more significant in the non-cancerous cell line HEK293 than that in the cancerous cell line 9L, suggesting its application in diagnosing tumor. Several other components also have MR signal at around minus 1.6 ppm. However, they are in mobile state and thus cannot produce a negative NOE signal. Here are take-home messages. First, NOE minus 1.6 is close to water resonance and thus is significantly influenced by direct water saturation effect, resulting in a dip that is not easily identified from the Z spectrum. However, it can be isolated using specific quantification method. Second, NOE minus 1.6 may arise from the NOE between membrane phospholipid choline head group and water proteins. Third, NOE minus 1.6 may be a new biomarker for diagnosing tumor. Thanks for your attention.